subscribe to our YouTube channel, Joy Learning TV.
Hello, our lovely viewers, students, and all watchers of SHSR Joy Learning. You are welcome to the biology section with me, your facilitator, Osei Kwame Amoateng, going to take you through a Form 2 topic in biology titled Phyla of Fungi. We are going to explore the exciting world of the fungi, something that is so common to us in our homes, in our bags, in our shoe racks, in the medical world, a lot of use of fungi. You'll be surprised to know diseases that affect you, and even our ladies will explore something. So gather yourself, find your pen and paper, and take some notes, and try to relate to things that are around you. Today's topic, at the end of this lesson, the special objectives we want you to have is that you should be able to list the major phyla of the kingdom fungi and describe the major characteristics of such members of the kingdom and the phyla it belongs to. If it's possible, I'll show you pictures. We assume you are watching it under the microscope. Try and see if you can sketch or draw appropriately some examples of these. We also take our time to explain why they have some names. If you read different books, you may come across more than these three on your slide. Per ISS, we said the main or the major phyla means that there are others. In some books, you come across 5 or 6. Others, you come across 4. But for our SS, let's keep to this. There are 3 phyla of the kingdom fungi, of fungi. The first one is the Zygomycota. I hope you should be able to mention it. Then you see in bracket Zygomycetis. When I'm trying to just talk without mentioning, say members of Zygomycota, I say Zygomycetis as the name of the organism, as you say, verb and noun. The next is Ascomycota, and the last is Basidiomycota. The names all come from some Latin words depending on the structure of their fruiting bodies. So now let's begin by taking the first one. But before I even go to the first one, let's look at the general idea of fungi. The kingdom fungi. I believe you've seen things growing on your bread, your shoes, even on oranges, our favorite mushroom. You've seen woods being decayed and you see some things growing out of them. You've seen ringworm on your head before. Probably our ladies get white, what they usually call white. All these as a result of fungi. Now let's look at what distinguishes a fungi from the other kingdoms. We started with the Protista the last time, and we said also that the plant group have cell wall made of cellulose. They also have storage material being starch. Formerly, because of how this group used to behave, we thought they were plants, because a critical look at them looks like they settle on something, grow some penetrating structures into it that looks like roots, and they spring up straight that looks like stem, and you see something crowning it that you may think was leaves or branches. But going to a little bit deeper, analyzing their characteristics well, comparing to the plant, we saw a lot of differences. For example, the plants have cell wall made of cellulose. Organisms of fungi rather have cell wall made of chitin. The difference is just something small. In cellulose, it is one glucose added to another glucose in that long chain, and bonded in between fibers which are laid parallel, where there's connection between one parallel line and another parallel line, making it strong. But in the chitin, or chitin, as some will call it, though they are laid also glucose in a lane, one aspect of the glucose is changed to have an ammonia or amino group in it. Because we add an NH3 in it. We say the ammonia group in the long chain. So each N has an ammonia group in the long chain, and that ammonia at the top bonds with the next layer down. We'll take a picture of that to later. Now, because of this, the cell wall is no more cellulose, but rather chitin. Plan to store excess food as starch, which we all know. We go and take starch of cassava, yam, and stuff. But when they get food in excess, they store it as glycogen, like the way human beings store theirs. So because of that, they can't be like plants. They can't make their food. So they are heterotrophic. They need to depend on already made food. Whilst plants are also autotrophic, they have chlorophyll to make their food. What they may look alike in terms of cell structure is that they all have large vacuoles. It kept me saying that they don't look like plants. They may say then they should look like animals. But they can also look like animals because animal cells don't have a cell wall. Animals move around. These organisms don't move around unless it's being swept away by the current or the wind. So you can't place them in plants. You can also place them in animals. So we needed to group them into a different kingdom. So formerly and mistakenly we were putting them in plants. Then we saw that they were not like plants because they don't share the same thing like plants. But they just look in structural form like plants. They can be animals because of the characters of animals they don't have. So scientists decided to put them into fungi group. As fungi like the higher kingdoms, they are also eukaryotic. That means all their cells, the organelles in them have membrane bounds around the nucleus, membrane bound around the mitochondria, a cover around the Golgi body, a cover around the endoplasmic column, a cover around the lysosome, a cover around the vacuoles. So based on this, we say they are having eukaryotic cells. Beyond the cell membrane is the chitin. In this group, most show multicellular or multicellular organisms. Few show some unicellular behavior, like the yeast. Then there are those that, though multi or whatever, they are so tiny that your eye can't, by its naked nature, view it, see inside, or see into details, things that are inside, unless we use the microscope to study these things. So in view of that, some are microscopic, some are bigger that you can use your eye to see them. So on your screen is your chitin. Then they look like tube-like structure, tube-like threads, but they can stand because of the chitin that makes them strong. They can stand up. So like this pen is tubular. If I remove what is inside, it becomes a tubular structure. This is the tubular structure that enters the substrate. It enters the substrate and it spreads down as roots. One only tubular structure is called the Haifa or Hypha. But if plenty come together, then it is called the mycelium. When you have one, we say Haifa. If you have two, we say Hypha. But a mass of them, a fluffy nature, like plenty things around them, is called the mycelium. Let's look at an infection of a tomato on your screen. The white fluffy thing you are seeing there is the mycelium. it is saying that a person is an individual, but when we come together in masses in the community, we are saying we are a group of people. And we'll give maybe name to it. So one is a Haifa or Hyphae. Plenty things that you are seeing on the screen is your mycelium. So let's look at, let's say, example of one of the Haifa. This is using a Zygomycota group. You can see on your screen this thin thread tubular structure that you see there, which is a thread-like material, is called the hyphae. If 10, 20, 30, 40, 50 come together, then we are calling the mycelium, the fluffy nature of it. In addition, anytime they want to feed, I told that formerly we thought they had roots. But the last ends of the Haifa that comes up, the Haifa is the one that comes up. The one that go into the substrate, the down part here, are called the rhizoids. These rhizoids enter your bread, your kenke, your whatever. Then at the tip of the bread, they will secrete chemicals. Okay, let me pause there and explain it the other way for it. When you eat, the food enters your mouth, goes into your stomach. You secrete enzymes onto it. The enzymes will break down the food inside your stomach before we will get the nutrient and absorb it into our cells, which will help us grow later. So to get the nutrient, it went into you, you separated them, left the chaff in your intestines, which will bring out as fecal matter, and we take the nutrient part. In rhizoids, they don't do that. Sorry, in fungi, they don't do that. It's like, don't let it be nasty, but assuming there is enzyme in your mouth and you spit onto a bread, then the enzyme will just break the bread into parts, the nutrient part and the waste part. Then you just take a straw. Then you take only the nutrient part and leave the waste part behind. So what happened? The saliva came from my mouth onto the bread outside my body. The digestion is taking place on the bread outside my body. So we are saying that digestion is extracellular. It's beyond or outside our body. For the one I described like you put it in your mouth. It is inside your body, intracellular. And even the proper digestion takes place in our cell. After we got to the nutrient, the proper digestion takes place in our cell. For them, the digestion takes place outside their body. So the bread, if you see them growing on bread, though they are in the bread has not entered their body. They rather secrete enzymes onto it, break it down, take the enzyme, and leave the bread down as if the bread, nothing has happened. By the time you touch it, it's spoiled. So their digestion is called extracellular, and we term the feeding relationship saprophytic form of feeding. Others are also not saprophytic, they are parasitic. That means they parasite on your body. They get hooked onto your body, then take nutrient that you have already digested. This and they will not digested by you, then they just take it in. They are parasitic, like the ringworm and the white our ladies get. Now, fungi are found growing on damp, moist, decaying organic substrates. So what they grow on are called substrates. It should mostly not be living. It should be dead or it should be damp or something. So you understand why fresh bread is not attacked. But when you put the bread in the rubber for some time, some days, and you don't keep it in the freezer to reduce the temperature at which their enzymes work, then the bread will get spoiled. Because some heat will be formed in the rubber, causing the bread to also decay over some time. Examples of the general group which will be taking our time to classify them was the Rhizopus, the mucor, our favorite yeast used in the bread, the claviceps, the bracket fungi, the mushrooms, etc. So we take them one after the other. Let's look at the first group called the Zygomycota group or Zygomycetis. This is the popular common one we find. Your bread, it's on. Your kinky, it's on. People are fan of when you see some white mold or some black, and then you cut it away quickly. Then you say, oh, I've taken it away. And you can eat it. You can just, your shito, student at SS, you see something growing around your shito, some white thing growing there. You are saying you've been attacked by, right? We can't be conclusive, but usually they belong to the Zygomycota group. How do they work? First, they are unicellular and filamentous forms. There are those that are unicellular in them and then filamentous. Now, when I spoke about the Haifa, there are two types of Haifa. On your screen is the image I showed you first. You see that the tube is long, and I've not put any cross walls in between. Now I'm putting something that separates the tube into components. If I should do that, that means I'll form a cross wall in between them. Then we say the cross walls are called septate or septum. A septum is a layer created by the tubular structure, but they don't end. There is a space in between them. So there's a small hole. Now they divide one region from the other region so that this place can have its nucleus and content slightly separated from the down. But if it is tubular, it means that the nucleus are just in, running up and down. Cytoplasm is in, running up and down. So for the group of Zygomycota, their hyphae are non-septate. There are no cross walls. So it's like a tube with water and nucleus are passing through. I'm just using the water to symbolize cytoplasm. And the nucleus is passing through up and down. Then I'll show you a picture for the third point. They are lined with the tubular hypha with lining of cytoplasm containing many nucleus. As I said, I shall have divided them into separate cells so that everybody can have one or two nuclei. But this is the case, the wall is not there. So what's supposed to be in one, and now we are seeing them in this whole thing. So there are supposed to be 10 cells here, everybody having one or two nuclei. We are going to say, we are going to have about 10 or 20 nuclei. But now they are all in this long thing. So we say that then a tube is full of nuclei. Let's take a picture of what I'm trying to explain. So take a critical look of the Zygomycetes picture. The inside is all this inside is the vacuole, a big vacuole. Surrounding the vacuole is a thin amount of cytoplasm. And you can see, let me say 1, 2, 3 nucleus scattered in that cytoplasm. So this side is your cytoplasm too. Then beyond the cytoplasm is your cell membrane. And beyond that is your cell wall indicated here. So that is the image of a Zygomycetes hypha. If it was a septate hypha, I'm just trying to use another color. If it was a septate hypha, we would have separated this portion from that so that there are about 3 cells you can see on your screen. But it's not that type. Another point you are supposed to know. Okay. Let me show you an example of a real Zygomycota. The type I'm going to use is the Rhizopus group or species. I believe you've seen this in the house. This growth. Yeah, we cannot be conclusive very well because some can be in the Ascomycota group, but majority of the bread is the Rhizopus. At SS, we use Rhizopus. Technically, there's a difference between a bread mold and a Rhizopus. But this is an example of what grows on the bread. So what you see growing on this bread, before I end, ask yourself what is in the bread that is making them comfortable, conducive to grow on, so that when you know that, you can prevent them from destroying your food. If it was in the lab, we'll take a sample of this and put under the microscope on a slide and covered with a cover slip. You will love the amazing picture you see. A silver-like thing with threads like skeleton passing with round tops. It's very beautiful. Very beautiful. Then you see how they penetrate your bread. So this small thing, you can see at the back, it's penetrated to this side. So how will I know how far they've gone and how far their enzymes have penetrated the bread and destroyed the bread? And they are all eating from each side. So before you realize, a chunk of this area, the outside has been eaten. So this is an example of a natural Zygomycota. Now you see this black dotted thing, means that it's matured. The dot is not the entire structure. The dot is, the black color you are seeing comes from this tip. The tip on your screen. So this area is the black side. That area is called the sporangium or sporangia. It is the place that houses or make spores. These spores will break. For the process of making the spore, because they didn't need another fungi to reproduce with it, we refer to as an asexual reproduction. Then they will make spores by a method of meiosis, where a nucleus will divide into 4 smaller, unidentical forms. Once they become plenty, they will burst and spread in the air at your kitchen. So you understand that you clean, you mop, you do everything, but they are in the air. Hardly do most of them settle on your table. Whenever you dust something, some will fly. Maybe you put your food and don't cover it. You expose them. The next 2 days, they've attacked something. Either your kinky, your bread, your something at the kitchen. So the question is, think about, I've asked you first question, what conditions are in this that makes them, then which ways can we deal with them in our kitchen? Since they be carried by air. So what we saw, the black top is the sporangia. Then when they want to reproduce sexually, they use a method called conjugation. So you see many are together. When they think conditions are going to be tough, example, after eating everything in the bread, and they know the bread has lost all nutrients, they can die. One Rhizopus with its sporangia at the top, and the sporangia, what holds them is the sporangiophore. On your screen, I've underlined sporangiophore. The rhizoids that were digesting the food, they are those that grows horizontal, not vertical. So the rhizoids grow vertical into the bread. Those that faces each other horizontal are called the stolon. They will meet and they will share nucleus together. The nuclei will fuse, and when they fuse, they will form a wall around it. And in biology, when 2 parents fuse, what we get is a zygote. So we get a zygote, and once they make a thick wall around the zygote, then we call the zygospore. And it's from this that the name Zygomycota comes from. The way they fruit and make new organisms is called the zygote. So their maturation and producing new one is called the zygote formation. So we call it zygospore. So on your screen, I'm going to show you what I just showed you, the bread. An example on your screen. And one they've cultured in a petri dish on your left. So if your teacher take you through this in school, this is what you are going to get. Then you just scoop a little and put on a slide and view under the microscope. So it's not healthy to eat things that have been decayed by Rhizopus. So a typical study under the microscope, this is what you will get. If you can isolate one, you see the wall of the sporangium, the sporangiospore. The spores inside, because it's formed the sporangium, it's called sporangiospore, or we just call it spores. We'll come to columella when we study Rhizopus into details. There are no septum, there's sporangiophore here, and you have seen the substrate mycelium. There is a picture of what happens during reproduction on your bread. So what I show you, this is taking place. What I just showed you on your screen is also taking place, which you can't tell. But those that are black, I said, are already matured and probably ready to break and spread new ones. It's a topic in the life process of organism, so we'll come to it. Now, conditions that ask you that favors the growth of mucor or Rhizopus. Now, the food item must be dead and decaying. Let me add the D there. And decaying. Stale or spoiled or uncovered. So, one, always try to cover things at the kitchen. Don't expose them. The food item must be wet or moistened. So if you can usually put in the dry, you have a way of keeping it dry, they can't attack it. That's why some people leave their food in microwaves and stuff. After heating, they keep it there for a while. The food item must be in optimum temperature. Their enzymes work at a certain temperature. So if you can find a way to destroy that enzyme from working, when we know that enzymes are affected by temperature, you can reduce it. So you put it in freezer to reduce the rate at the enzyme work, or you heat it to reduce the rate or destroy the enzyme ability. So it needs carbohydrate, protein, so you have to make sure they don't get this carbohydrate to live on. The next group, resuming to the next group on your screen, the Ascomycota, called the Sac Fungi. Let me show you examples before we go on. This is not actually why it is called the Sac Fungi. This is why we call it the Sac Fungi. Sac. You know how we put things in a sac? Sac Fungi. That is the Asco Cap. I will be studying how the sac is formed. So for them, the Ascomycetis are most of them are multicellular, except usually the yeast or Zygomycetis. The yeast group are unicellular. They are, apart from the ones they are unicellular, that is, the whole organism is in one cell. They can have septate, but the natural group of Ascomycotas, they are Haifa that is here on the board, as I'm putting some lines in between them, explains that each tube is divided by septum, so we say it's a septate Haifa. Yes, a septate Haifa. This crosswall is not continuous. There's a small hole that allows one cell to stream its content and pass some to the next. So when I take food from the, let's say, assuming the bread, I can pass it from one cell till it gets to the top because there's a little hole that helps in the transfer of nutrients. The next is that they reproduce in two ways. As I said, in all fungi, there are two forms. We saw in the first group, they can form by forming a zygote in a process called conjugation when conditions are bad. Or the tip that we saw, the black tip I showed you, where the sporangium, they reproduce without involving another person. It's asexual, and we say it's spore formation or sporulation. Here too, they can decide to reproduce asexual. Now watch your screen. The asexual are these small round things on the board. You see them as something with like feathers. It looks like feather. One strain. Then we have round things there, each called a conidium. So the whole thing holding them is called the conidiophore. Then the round things at the end is called the conidium. And in it are the conidiospores. That means spores produced in that small round things are called the conidiophore. So we say asexual reproduction involves spores called conidium or conidia formed at the end of specialized branch called the conidiophore. Then the conidia, which can call conidiospores, are the small spores that are made. So these holding structures, conidiospore, conidiophore, then this round thing is called the conidium, the container. With this understanding, we have those situations so that they can form sexual reproduction. Now let's take our time to understand that sexual reproduction process. That's where their name comes from. Now when a spore falls on the ground, they will grow. Different spores will fall on the ground and they form threads, different types of them. Then they will start to pair. When they pair, they form septum. Because they are already in septum, two nuclei goes into the other will form two nuclei. And when plenty of this formation takes place, there will be a group of this formation or thread or hyphae that will form on the outside. We call them the sterile type. Another group form here at the start is the protecting one. So there will be protecting one around it. Then the middle group will keep having nucleus in them. When they feel they've reached a point that they need to reproduce, the cells at this tip here will decide to form a sac like this. And in the process, the two nuclei which were not fusing will then fuse over here as one. Once they fuse as one, then they undergo meiosis. So most time, all fungi body are haploid. Among the groups, they are the only one in the haploid body. Because immediately they form diploid, they will go back to meiosis and form haploid. Then they can form spores and fall down. So this structure actually that forms the sac is called the ascus. But because some were sterile going around it, it will form a whole cap-like structure called the Asco Cap. So the sac in which they form is the ascus, where the name comes from. That's the sac group. But the cap is called the Asco Cap. So they have this appearance of cap nature. But when you go deep and look at the individual hyphae that forms at the tip, where we make spores and burst out by its sexual reproduction, then we call the ascus. So you see that in the Zygomycota, we use the sexual reproduction phase process called the zygote formation with a thick wall around it called the Zygomycota to give the name to the group. Here we are using the ascus formed during the sexual reproduction to give the name to the group. So whenever we say Ascomycota, we are talking about sac-like fungi. So as on your screen, we say male Haifa called Cametangia will fuse with female called Ascogonium, leads to the formation of a productive body called the Asco Cap. So another picture, there is the Asco Cap or the top here. Then ascus are formed at the tip of the Asco Cap. Back to the picture. So I showed you that inside this are tip. You can see these lines coming up as tip ends where we form the Asco Cap. And the spores in them is called the Ascospore. So you add the name spores to it, Ascospore. A typical example is the Penicillin we have. Though you saw bread being attacked by Rhizopus, sometimes they can also attack this with different color of blue, green, sometimes on your leather. Some will argue whether this yellowish thing could be it. But a common one is the orange. When you see orange with some white formation, fluffy thing that when you touch makes powdery on your hand, then you are talking about the Penicillin group. You have shoes too that are affected by that. If you have shoes you place somewhere, leather shoes, and you go and it's damp and you see white things on, trust, mostly it is your Penicillin. Now let's take a time and look at the in-depth picture of Penicillin. So this is the asexual phase. But if they are forming the sexual phase, then they will form this. So this is the asexual phase, which I showed you at the top here. Now, a brother of the Penicillin is the check the way it's showing on this typical Penicillin. I didn't bring that picture here, so I won't mention it. Now let's look at the yeast too on your screen. I mentioned yeast in this group. You see that yeast is an individual cell on your screen. It's one cell, but the cell can bud. A bud means that a portion of cytoplasm from the parent will pass into this. The nucleus will divide into two, and one will pass into this young one. So the original parent here has divided into two and given one to the budding cell. Later, when this one matured, they also divided into two, gave one to this budding cell. So the process of an outgrowth formation with cytoplasm pass into it, a nucleus passing, for it to become an individual organism, which can cut off from the parent, then we call that process the budding. Yeast is so important because of its role in our life. The structure of yeast, as I described, is just a cell with a cell wall, which we saw it was made of chitin, little cytoplasm, some nucleus in it. And then their unique role is that they can respire in two ways. Their respiration depends on the presence of oxygen. If oxygen should be there, then they'll just break down their sugar content into carbon dioxide and water, the normal respiration. But usually, they can also find themselves in conditions that there's no oxygen. They prefer that one. So we associate the situation where they don't use oxygen. What happens is that when they don't use oxygen, they'll use a process called fermentation to break the sugar into alcohol and carbon dioxide. Again, into alcohol and carbon dioxide. The enzyme within their body that's able to take that process and form the alcohol and carbon dioxide is called the zymase. Underlined on your screen, zymase. Now, what they do is that it's a complex glycolysis process. They don't enter into the Krebs. They hold on to the glycolysis process and go to the next process we call the alcohol fermentation. So when you have yeast put in bread, you see that they would make sure the bread has carbohydrates. They will add sugar, they will add whatever they will need it. Then they will put the yeast in it. Then the yeast will feed on the sugar or the carbohydrates. And the process, they will break this down into the alcohol and the carbon dioxide. If you eat it raw the next day, if it's plenty, you feel this sour or alcohol taste in it. But once they put the whole thing in the oven or the furnace to heat it, you find out that the alcohol, which is volatile, will just escape. Boom. But the carbon dioxide that want to escape would try to stretch, and because the bread nature is that way, it will try to stretch it and make the bread bigger. So you see most guys buy bread and they just do that. Means that they are trying to take away all forms of carbon dioxide that is in it. This same idea is used in the brewing industry. You put sugar, you put yeast, you cover it, no oxygen, it would break it into alcohol and carbon dioxide. Then they will distill and get the alcohol. So on your screen is a picture of yeast. The whole thing, the yeast is so tiny, but we've just put it together in a bowl and taken a picture of it. If you have opportunity to see under the microscope, it's a little bit longer, like a capsule. Very beautiful. Another example is the morel, which is also an Ascomycota group. This one is eatable, so people eat morels. One important thing I'm supposed to know is that I've said about yeast, very important in our life, making alcohol and bread. Penicillin is used usually in drug manufacturing on yeast, cheese, in making cheese, and also in making drugs. The penicillin, that white capsule that we take when we get infection, is penicillin. We have the Candida group, the ones that make it get white. So you have the Zygomycetis, you have the Sodari group, that cow dung, they help decompose cow dung. You have the Aspergillus group, that causes respiratory disease in our humans and other organisms. Then most of these groups also form lichen with protista group of chlorophyta. Then the Candida species, yes, the Candida, usually, girls suffer, even only girls, men can also get white, shockingly, but men can also get white. But most of the white that are young ladies or mature ladies sometimes get as a result of infection is also from this yeast group. Say yeast, from this Ascomycota group. Now let's go at the last group called the Basidiomycota, the club-like fungi. They are usually the biggest you mostly see around. They are in the mushroom group you've mostly seen. Now these ones usually live on decaying wood with a lot of cellulose in them. They are thing that enters is called the Haifa, but this Haifa, like the Ascomycota, is also divided. So we say septate Haifa. There's a unique behavior of this group. Not like the bread type that once they fall, within a short time they grow up. Mushroom can take years to form underground without coming up. But the day they want to come up, it's so beautiful. We take our time and look at them. But let's take some pictures. The one on your left is your mushroom, and the one growing on the cat tree, the stump there, is the bracket fungi. Maybe you've seen it somewhere before. Take a time and look at it. We have the toadstool too. That sometimes reddish, grows on logs that are on the ground. You sometimes use your leg to push against it and it falls. So let's go back to Basidiomycota. So now that you've seen what's on the board, let's take a life cycle which makes it easy. So the mushrooms start on the ground with the primary Hypha. And the primary Hypha of different types intertwine and form a thick thing that looks like a stem. We call it a stalk. So one stalk has plenty different types of Hypha intertwine against each other, forming like a thick rope. Then once they get to the top, they form a bulb. Then the bulb would open up and it's called a cap. So this is the cap. Now, when the cap is ready, under it are some lines. When you look at mushroom, you can just nicely pass your hand over it. It's called that umbrella, like the down of an umbrella. It's so smooth. We call that thing the gills. On the gills, as you see on your screen, on the gills are these lines, and on each line is this structure called the basidia or basidium. One is called basidium. Plural basidia. It is in this that we have nucleus of different hypha coming to join together in it. And when they fertilize and they are ready to undergo the asexual phase after fertilizing, they will form this club-like structure, club-like like this. But when we go to the ascus, it looks like just a sac. So the name club-like in the Latin is the basidio or basidium. That gives the name Zygomycota. So just for you to appreciate it, the way we get the name. So the basidium that we say possesses a reproductive cell called the basidium under the cap, which you've seen the cap now. Then the fusing of the basidium leads to formation of the basidiospore, the spores that they would release from the club-like structure. When they fall down, they will take time to penetrate the wood or the soil or anything that is decaying in the soil, usually cellulose product like wood. Then, after years or months, they will rise up again and form. So before you pull one mushroom off, remember that the legs have spread some distance away from where the real mushroom is growing from. So example are the mushroom itself. You've seen the bracket fungi. The toads too, I couldn't get to an example. Then the rust and smut are basidio group or fungi group that causes a lot of disease to plants. Now that you know these three groups, same pictures, seeing why they are called basidio, because they form basidiospore or basidium and stuff. Ascomycota, conidia with conidiophore, then the ascus and the Asco Cap. The Zygomycota, zygote formation with a thick wall called the Zygomycota with a wall around it called the zygote. There is some benefit or let's say importance of this group. Even before I end, you know economic importance does not mean only good. Bad is also economic importance in biology. One of them is associations they form with other organisms. And the common ones they form is the mycorrhizae and the lichen. A mycorrhizae is a relation between a fungi group, usually the Ascomycota group, with the roots of plants. So you have your plant this way with roots growing down. Every root has a normal root hairs that can allow you to take water some distance away. But if they form the fungi are thread-like and they can move far from the roots. So they can go some distance where the root itself cannot reach to tap water. And these ones can tap water for them. They can break down substances and absorb the nutrient and give it to the plant so that the plant can grow. So association between the fungi and the root of plants, some root of plants is called mycorrhizae. Association between fungi and some algae groups we study under chlorophyta is called the lichen. So a picture of lichen is on your screen. So this has two groups, a fungi in it and the algae, like the Sporangium you see around, in one group. I'm not saying Sporangium, but because Sporangium belongs to chlorophyta, I'm drawing your mind to it. They are also bioindicators. They are used as pioneer group in succession on bare land, that we also call the primary succession. They are the first to break down substances there. Other examples, other importance we can also maybe write down are that we've seen that they cause diseases like candidiasis, they cause diseases like athlete's foot, also ringworm on our head. We've seen mushrooms serving as food. We have seen some like rust and smut that can act as pests and destroy plants. You've seen Penicillin and Co. and Rhizopus that can attack food and destroy it. So these are some examples that we can write down. They also can decompose things, because when a tree falls down, they will eat. Let me use the word local, like literally eat. Because they are eating, and the process of their eating, they will break down the nutrients, the whole log. When they take what they want, the parts that they've taken the food from, they will break the molecules. It will break the whole molecular structure so that the molecules does not hold, because I've taken the nutrients, which are also molecular, away. All of a sudden, the whole log will drop. So the nutrients and the molecules that are in this food, because they take what they want, the structure becomes weak and they fall apart, and we are saying that they've decomposed the food. And they've returned the nutrients to the soil. If they're on the soil, they'll go back to the soil, and plants will use them to grow. So we say recycling of nutrients. Plants make food, the food we eat some are stored in. When they die, it goes down again, and the cycle continues. There is this fourth group we don't mention in SS. We call them the Zygomycota. We call the imperfect fungi. We call them imperfect because all the three I explained, they have both sexual and asexual phases. Now, imperfect fungi are fungi that, as of now, they've been able to only discover 8 asexual phases. They don't know any sexual phase they have. So because of that, they didn't become a complete or a perfect fungi in the sense of growing, using one method asexual to reproduce, under bad conditions use sexual. But for them, they don't have the sexual. The only one we know is the asexual. And scientists have grouped them into one group called the imperfect fungi or Zygomycota. I hope we've taken time to pass through this group called the fungi. You've enjoyed it. You've now become aware of how they grow on things, maybe your kitchen, your textbooks, if it's leather, nature, your shoes, your bag that are leather. Our ladies have also come to understand that candidiasis is a sort of fungi, not any other form. Ringworm we find on our head are all this group. Our bread, we know it now. Orange, when it's getting spots, we know it. Tomato, sometimes also we have this white thing growing there. All these are the fungi group. We've seen their role in our ecosystem. So to bring our lesson to an end, let's summarize it with these 3 areas. Fungi mostly possess hypha and have a body called mycelium. They have saprophytic nutrition that leads to decomposition. And they reproduce asexually by spores and also sexually. So before I go and we meet another time, I want you to try to test your hand on this past question that has come in various forms, especially in practicals. We mostly bring this in practicals a lot. State 4 characteristics of kingdom fungi. State the 3 main phyla of fungi, and in each case give 2 examples each. Then tell us 4 importance of fungi to man. I hope today we've made it easy for you to understand. Love this organism. Don't be afraid of it. Just mostly wash your hand and keep your food in the freezer or heat it. To come your way again the next time, I'll be your facilitator, Osei Martin, taking you through phyla of fungi on SHS Hour on Joy Learning. Hope to meet you once again or our next lesson. What can I say? I say bye-bye till we meet again. Bye.
Subscribe to our YouTube channel, Joy Learning TV.